So welcome to PCM4U podcast. Today I want to discuss with you a topic about artificial intelligence for clinical cancer research. So we will see today that virtual screening can increase the number of lead pharmaceutical compounds. Then that AI can predict the likelihood of treatment approval from early phase clinical trials. And then at the end, we'll see that the recruitment of patients in early phase clinical trials can be improved by NLP techniques, which stands for natural language processing. So first, I want to describe to you the different steps of pharmaceutical development in many areas of medical research and specifically for cancer research. So there is a step of drug discovery where the aim is to discover a new compound. Then there is a step of preclinical screening of the compound. And at phase 1 of the clinical trial, this is the first time a human takes the compound, and this is dedicated to analyze the safety of this compound in the humans. Then there are two other steps, the phase 2 clinical trials and phase 3 clinical trials that are used to confirm the effect of the compound in patients. So at different steps, there are implication and development of artificial intelligence tool to improve either the quality or to increase the output of each steps, and we'll see their impact. The baseball player stands here to represent that. In fact, this is clearly the early phase of the the drug discovery, preclinical screening, and phase 1 that are detrimental to increase the chance for a compound to be approved at the end. But the final approval often is decided on the results of the phase 3 clinical trial, which is a confirmatory clinical trial. So let's go in each of the phases. First phase, I want to describe to you the different space that we have behind us. First, the chemical space and the protein space. Protein space because most of the compounds are targeted to a protein which do a function in the human body. And most of the disease are dysfunction of some proteins or some mechanisms. In the human cell, we have more than 20,000 of proteins that are encoded in the DNA. And for those proteins, in fact, only 2,700 are drug targets. So you see that there is room for improvement to find new targets in the protein space and develop new compounds. For that, it is important to know the structure of the protein, and there are different ways to analyze the structure of a protein. So the classical way is to make structure analysis by microscope, for example. And there is a new way recently improved by AI to predict the structure, the 3D structure of the protein from the sequence of the amino acid of the protein. And this clearly opens the doors to the analysis and the prediction of the 3D structure of all the proteins in the human body, either the normal ones or even the altered ones that relate to some diseases. So this is for the protein space. And then, in the other hand, we see here the chemical space. Chemical space are all the pharmaceutical compounds that we could think about. So you see here in the graph that we have 9,600 compounds that are called drugs here because we know their effect on the humans. And there are databases for all these compounds. For example, drug bank is a database where we can find also descriptors of those compounds, those treatments. But then, in fact, we know the structure and some of the activity of compounds for 92 millions of pharmaceutical compounds. For those, we have less descriptors. For example, we lack the descriptors on the human, like pharmacokinetics, pharmacodynamics. But we know, for example, the bioactivity. And all those compounds, we can find it also in public database like PubChem. And it has been used a lot to develop AI tools, such as the one for virtual screening. So how does it work? Virtual screening stands on training a model to predict the bioactivity of a compound based on its chemical formula, mostly, and the target proteins. You can train on these 92 millions of compounds and even more pairs, and then you obtain a model that could predict the bioactivity of new chemical structures that leads to and opens the doors to explore even unknown chemical structures. And this is what is represented in the big bubble in the chemical space. In fact, it has been estimated that there are 10 out of 62 potential pharmaceutical compounds given all the possible pharmaceutical structures. And virtual screening is currently exploring this space to provide us with new compounds targeted to altered proteins. So the preclinical steps of drug discovery has changed because, as you see in this graph, the classical step of manual target identification up to library development, which used to take a lot of time and still for some situations take a lot of time, can be clearly improved, and the output can speed up by virtual screening with the methods I presented to you. Then the next steps are still in the classical ways, such as high throughput screening, lead optimization, and then preclinical and clinical trials. And we'll see now what AI can do for high throughput screening. So preclinical screening stands for analyzing the effect of the compound in, for example, for cancer research in cell lines, in cancer cell lines. We have plenty of cancer cell lines, and the aim is to evaluate the effect of different doses of compounds on those cell lines and evaluate their anti-tumor effect. For that, we also have big databases, such as the GDS database, but there are many others. And I wanted to show to you this paper that clearly change the landscape of AI applied to predicting the drug effect in cell lines. So the authors used the data of almost 1,000 cell lines and 200 drugs, and it leads to more than 100,000 pairs of cell lines and drugs. So it's a big database that you can train models on. So this task is a long-lasting task. It's not very new, but the first generation of models were quite limited in terms of performance. And then came some improvement in the architecture and the data used to input in the model that allowed to increase the performance of the model, and I will show it to you just now. First step, to explain a little bit of the model. The authors choose to use the gene expression from the cell lines and the gene expression only. So why did they choose that? In fact, there have been previously a lot of literature of evaluating and comparing the different omics that are the most performing to do this predictive task. And most of the authors agreed that the gene expression is the omics data that is the more related to the drug effect. So the authors now just restricted their input data to gene expression, and at the other end they took the representation of the drug in the format of SMILES. So SMILES is a format to represent drug formula or pharmaceutical formula, and you can see an example here in the picture. And it is basically characters, sequence of characters. So those two types of data, gene expression which are numeric and continuous, and SMILES which are characters, can be inputted and transformed in numeric formats, and then were stacked here in many layers of convolutional neural nets. And at the end, the label to predict was the IC50. IC50 stands for inhibitory concentration 50. This is the concentration of the compound that kills 50% of the cancer cells. And it represents the effect of the compounds at the end. So that's what the authors wanted to predict. And you will see the results just now. So here are the results on the test cohort. So the test cohort is, or the test set, is a set of data that the model hasn't seen for the training. And here we see in the y-axis the measured IC50, so the true value, and in the x-axis the predicted IC50 from the model. And we see clearly a great correlation between the two values, meaning that the model predicts very well the IC50 from the compounds that it didn't see just before. Pearson, for example, Pearson correlation was 92%, which is a very high score and really changed the landscape of predicting drug effect on cancer cell lines. So now that we have AI at the drug discovery phase and AI at the preclinical screening phase, are there solutions that do all in one? And I want to show you a single example. It's a company that had a first example of going from the target protein up to a compound that started phase 1 clinical trial, so the first administration to humans. And it did it based on many different AI or numeric techniques. And what they show here is that thanks to those methods, they really speed up the different steps. These steps usually took more than 2 or 3 or 5 years in the classical way, and here we see that they took 18 months to go to formulation and manufacturing, and then this formulation and manufacturing step took 9 months. So they did their first in-human clinical trial in less than in almost 2 years after the identification of the compound. And all those steps that you see here, the small cubes, in fact represents some AI techniques, as I showed you, to discover and to identify the lead candidate compounds, to evaluate their effect on, for example, cell lines or in vitro studies, and to predict their efficacy in the clinic. So this trial has started, I think, 1 year ago. I haven't seen to date some results, but we are really waiting to see the results. It was dedicated to a patient with pulmonary fibrosis, so it's not cancer applications, but it is, I think, a great example of applied AI methods for drug development. So now let's start this clinical phase, which comprise phase 1, phase 2 and phase 3 clinical trials. So the main problematic in this phase is that there is a very big attrition rate, meaning that in cancer, for example, in cancer drug development, 5% of the compounds that start a phase 1 clinical trials will finally get approval. So you see that this is a very low rate, so there is a high rate of failure during this clinical trial phase. The failures are mostly due to either toxic compounds or non-efficacy. That are the main reasons. There are many others. And so improving the prediction of the efficacy or the toxicity of the compounds could help increasing the percentage of drugs that are efficient for the clinical phase and would lead to approval. So this graph represents to you in which phase of the clinical trial there is the biggest attrition rate. And we see here that from phase 1 to phase 2, there are almost 50% of the drugs that pass this phase. Then the biggest bottleneck is phase 2 to phase 3, where you see for cancer compounds in red, there are only 24 or 25% of the compounds, so 1/4 of the compounds that pass this step. This can be understood because phase 3 clinical trials are open for thousands of patients. They are very costly. They are long to run. So there is the decision to go to a phase 3 clinical trial with the compound is a big decision in terms of investment and exposure of patients. But clearly this is where we think we can improve the approval rate with models. And then you see that from phase 3 to NDA or NDA to approval, there are almost again 50% of the drugs that pass after phase 3 is ended and is complete. So how AI and machine learning models could increase approval prediction to select the best drugs to start clinical trial. So I want to show you 3 examples that have been published. You see that different pharmas are invested in these applications, and the first one from Novartis, and I think was in collaboration with the MIT. The aim was to predict approval from the phase 3, the results of the phase 3 clinical trial. They were provided with a huge database of phase 3 clinical trials and descriptions, and the approval or not of the compounds. And we see that the performance of the model in terms of area under the ROC curve, which represents the performance between 0.5 and 1, their AUC was 0.88, which is a very good results with only machine learning models. Then another initiatives proposed to predict phase 2 to phase 3 transitions. Where is the main bottleneck? And this collaboration provided a model that had an AUC, so a performance again of 0.88, which is a good performance, so it gives confidence that it's possible with machine learning models with data from phase 2 to predict what is the outcome of a drug in terms of transition or approval. And so we did the bet that maybe if we go from phase 1 results. We could predict the approval of the compounds, and with machine learning techniques, with the pharmaceutical descriptors and clinical descriptors at the end of the phase 1 clinical trials, we also obtain on a test data set an AUC of 0.89, which confirm that we can predict with machine learning models the approval of the compounds, and could help to increase the probability a compound starts clinical trials with a good probability of approval at the end. So my conclusion for this first part about pharmaceutical development are, I think, that I convince you that virtual screening and protein structure prediction allows the exploration of a huge chemical space. And the impact of that is that it could increase a lot the number of compounds that starts phase 1 or clinical phase of evaluation with patients. And then NLP techniques or machine learning techniques can increase the prediction of the effect of a compound from the completion of a phase 1 clinical trials. And so that it could help selecting the best compounds to go for the phase 2 and phase 3 that requires more patients. So this is the end of this part. I will see you again for the part on clinical trials in practice for patients in a few minutes. Thank you.